Hello everyone. So today we are going to see the task 2, that is generate a translation of DNA and RNA sequence into amino acids. As you can see, we have learned previously like how to download a biological sequence from the different databases like EMBL, NCBI database. So here what we are going to learn is we are going to generate a translation process of a DNA sequence into amino acids. So what does it allow us? It allows us to predict the protein sequence which are encoded by a specific gene. So this follows the certain central dogma of our molecular biology and as well as the genetic engineering. So in which it states that a certain DNA is transcribed into mRNA, which is further translated into amino acids or proteins. So, as before we begin the practical sessions, we have to know what this process is called. So, this process is called gene expression. So, as you can see the image, and it is very responsible for synthesis of all the proteins, all the amino acid in a particular cell. Okay. For example, the steps which involves in certain translation is which has a genetic code in the particular mRNA sequence, which read in a group of 3 nucleotides, as it known as codon. So each codon is correspond to some specific amino acid, and the sequence of the codon is always determines the amino acid sequence of our protein. So that's what we know as our DNA or RNA sequence of particular gene. So we can predict the particular protein sequence or amino acid sequence which are encoded by that specific gene. So here, how to how to edit this sequence through which software. Our first step is we are going to download the BioEdit software. So what it helps you is, it helps to view the sequence, it helps to edit the sequence, which you will need is write and read the FASTA files. This is the one of the such tool. So it is like free software tool that can be downloaded from the browsers. Just Google it, BioEdit, you will be getting that option. So by viewing and editing the sequence using BioEdit, as we downloaded this sequence from in the task one, as we remember. So what we have to open the FASTA file again, continuing from the task 1, and you can see the displayed sequence. So which we can use, there are different toolbars, different menu options, which you can navigate through the sequence and you can edit it and you can perform various analysis using BioEdit. So you can see the sequence menu. This is our step 4. You can see the sequence menu. Okay. Click on upload the sequence and go to sequence menu. You scroll down and you can see nucleic acid. Then click on to it and it will translate and you will get a drop down menu. So we have to choose the reading frame. So we have our DNA sequence, which is made up of a series of 3-letter codes. That codes are for specific amino acid or a specific protein. However, the same DNA sequence which has a different frames, so which lead to a particular different amino acid sequence. So don't worry, we will be performing the practical of the session. And the final step is we will be generating the translation. So the translation should be appeared like this in the new window. So with some amino acid sequence also being displayed below by the corresponding DNAs. So generating a translation of a DNA or RNA sequence into amino acids that allows us to understand the protein coding of the potential of the given sequence. So as a researcher, as a bioinformatician, as an analyst, we have to identify the potential functional domain of a particular protein sequence. So, for example, like if you are taking any type of diseases, for example, cancer, you are isolating a protein. From that particular protein, you have identified potential functional domains sequence, and you are going to work with the gene expression. And you are identifying how the translation happened from DNA transcribed in mRNA and further converted into protein. So it can provide some insight about the protein functions and structures also. And we can also develop, it also help us to develop some targeted therapies to treat the particular diseases. So now let's move on into the practical session. Now, this is called BioEdit sequence alignment element. So this software you can download it in the browser and you can do it. So what we have to do? Yesterday we have downloaded our sequence and we have viewed it by using the BioEdit sequence alignment editor in our previous task 1. So with the continuation of task 1, we are going to do the translation step. Now, what you are going to do is you are going to load your sequence. First, go to File, go to Open, so it is loading. So in the previous task, we have downloaded the sequence too. So same thing, we are going to use it for this step also. Just click, give open. So your sequence has been loaded. So what is the next step? So you can find the menu called sequence. You can just click on to the sequence and go to nucleic acid. And you can find the drop menu. In the drop menu, what is your next step? You are going to translate. And you're going to use frame 1. Sorry, we didn't yet selected the sequence. So what you're going to do is we are going to select the sequence first. Go to sequence, go to nucleic acid, go to translate and frame 1. Here you can see the translation step. So here is your codon and here is your amino acid, which is represented, say, in the second line. As you can see, it has been translated. This is called our reading frame. And you can also see this stop codon and start codon. So as we have done our translation step, that is, we have the particular DNA is transcribed into mRNA and it is further converted into our protein sequence, that is amino acids. So hope you understand the step. So now it's your turn to do the same practical say, right? And this guide, whichever I have showed you, will help you to understand how to generate a translation of particular DNA or RNA sequence into amino acids by using BioEdit. So with this skill, you can also able to analyze more and more protein sequence. And again, it will tell you the function and structure. So good luck, and this is your turn to do the same practical. And hope, did you complete your task? Let us know in the comment section. Please like and share it too so others can also learn it. Thank you.